With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, the question here says that you suspect your patient to be suffering from a bacterial disease. However, the number of bacteria in the patient's body is very less. Which method can you detect pathogens in the laboratory? Now here we see that during the initial stages of the disease, the pathogen content is quite low. So the conventional methods, that is the blood and the serum analysis, or for that matter the urine analysis, will not be able to suffice this. Okay, so you cannot detect it because the pathogen is already in very less content. So for this matter, the pathogen's gene of interest has to be enhanced. So here we see that even if there is one pathogen present, with the help of this polymerase chain reaction, which is the PCR reaction, we can increase the number of pathogens in the sample, right? So first we collect the sample and then we subject this to this polymerase chain technology, or the polymerase chain reaction. In the steps of the polymerase chain reaction, the first step is the denaturation, which is done at a high temperature to separate the two strands of DNA. When the two strands of DNA are separated at approximately 82 to 92 degrees Celsius, then there is annealing, which is the next step. During the annealing, there is attachment of the primers, which are short chains of oligonucleotides attached to the 5' end at both the DNA strands, and followed by this the third most important step is the polymerization. This polymerization step is also called as the extension, where now with the help of Taq polymerase enzyme, that is the Thermus aquaticus polymerase. This is a heat stable enzyme which is obtained from a bacteria, which is a RK bacteria, that is Thermus aquaticus. So this Taq polymerase will help in the extension of the DNA and form two copies from one copy of DNA, and this cycle can be repeated as many number of times to increase the amount of pathogen in the sample which we have collected. So PCR is the right answer for this question, so we can mark option number 2 as a correct option. Whereas somatic hybridization is not this procedure, it is the procedure which is done during the plant breeding. DNA fingerprinting is used for the paternity disputes, for other things like for the crime forensic sciences. So this is also not the right answer, and hybridoma technology is used to make the monoclonal antibodies. So this is also not the right answer. The only right answer to detect such a bacteria in a suspected bacterial disease patient is the PCR. So that is option number 2. Hope you have understood this answer. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.